Hello, this slideshow or this video represents a review of the New York State lab diffusion through a membrane, the one with the onion cell and also the model cell. You should only be watching this video after you've completed the lab and given an attempt at answering most of the questions. This is also a good way to review for the verbal quiz, in particular the last few questions of the verbal quiz. So I'm going to take you through what happened in each part of the verbal quiz, or I'm sorry, of the lab, so you have a better understanding of what was going on, so you're better prepared for the verbal quiz and ultimately the test that you'll be taking. So in part one of the lab, what you did was you created something that's referred to as a model cell. And you took a plastic bag and you filled it with two substances. Inside of the cell, you added glucose solution, which is basically sugar water, and you added starch. And starch is a larger sugar. It's a bunch of sugar molecules put together. So that's the first thing you should know about glucose and starch, is that glucose is a small, simple sugar, where starch is something we call a complex carbohydrate, or a more complicated sugar. And it just means that there's more sugar molecules stuck together to make starch than there is for glucose. Glucose is things that you find in, like, the sugar that you would put in a cup of coffee. Starch is things that you actually use to make the collars of your shirts stiff. You can iron them into your clothing. And also things like bread are starchy. So just an example of glucose and starch. Both sugars. Glucose is simple, starch is more complicated. Anyways, these are two things that we put inside of the plastic bag. We then took that cell, tied it up, and placed it inside of a beaker. And we placed around the environment of that model cell, we placed water, surrounded by water, and we placed iodine. Iodine is a starch indicator, which means that when iodine comes in contact with starch, it causes a color change. So this is an example of the model cell. We filled up the bag about halfway, placed it into the beaker, placed water and iodine on the outside, and then we let it sit and recorded our observations for a few minutes. And this is more or less what you should have seen going on. After about 15 minutes, or maybe you left it overnight, or maybe a couple days even, you saw that initially we had started with glucose and starch inside of the cell, iodine and water on the outside of the cell, and then things started changing. We saw, first of all, a color change happening inside of the model cell, and also, you may not have been able to see this, but glucose did get outside of the cell because it was able to diffuse out of the cell membrane. There was a higher concentration of glucose here, so it went from a high to a low concentration, and it diffused out. We'll prove that in a moment. Also, there's initially a higher concentration of iodine on the outside, so it wanted to go from a high to a low concentration, and that's what happened here as well. The color change we'll talk about, and the one thing that's not represented well in this model is actually you should have seen that your model cell was more full with solution inside of it than it had previously been. So what was going on here, right? There's three substances that diffused through the cell membrane. The cell membrane was selectively permeable, which if you remember means that not all things could pass through it, and what was able to determine whether or not you're able to pass through that cell membrane was the size of the molecule. So think about what I talked about glucose and starch earlier, and with how that relates to iodine. So glucose, water, and iodine, they all got into the cell or out of the cell because they're smaller molecules. However, starch is a larger molecule and it was not able to pass through that selectively permeable membrane by diffusion. Although it's not enough to know that these things pass through, we also need to know, how do we know? What evidence do we have? You guys are scientists, right? I'm not going to believe you if you just say these things pass through as a scientist unless you actually have evidence that proves your claim. So let's go through it step by step here, right? Now, for the most part, we can see that there was a color change here. And I told you earlier that when starch and iodine come in contact with one another, a color change occurs. And that color change is a blue-black or purplish color, as you see here in our cell model. So that is the evidence that we use to say that we know that iodine, which is this amber color fluid that we dropped into the cell, was able to pass through the selectively permeable cell membrane and diffuse through into the cell. How do we know? Because when iodine and starch combine, it caused this purple, black, blue color inside of the cell. The other piece of evidence that you should know is that starch did not get out of the cell. And how do we know that? Well, I just told you that when starch and iodine get together, they turn the purplish color that you see inside the cell. Well, how do we know that starch didn't get outside of the cell? Now look at the observation that you can make about the solution surrounding the cell. It's still that amber color. It's not blue-black. If the starch was able to get outside of that cell membrane, the selectively permeable membrane, all of this should have been black. But that's not what we see. So as a result, we can conclude, based on our evidence, that starch did not get out of the cell membrane. And I'm telling you that's because starch is a large molecule. Another piece of evidence, it's harder to see in here, but it probably was easier to see when you were looking at your cell model, was that water was able to diffuse into the cell. If you remember, the only thing we put inside of the cell membrane here was the glucose and the glucose solution and the starch solution, about halfway full. We didn't put water beginning on the inside of the cell. We only put the water on the outside. But if you looked closely at your cell, you probably noticed that it was more full than it was previously. And that's because water was able to diffuse through the selectively permeable membrane and into the cell. And as a result, the cell became more full. If you don't believe me, go back and check your cell again if you still have it. Now, the last thing we need to check is the glucose. Did the glucose get outside of the cell? Remember, we put it initially on the inside. How do we know if it got out? Well, for glucose, we had to do a special test using something called Benedict's solution, or glucose indicator solution. And just like starch indicator solution, or Lugol's iodine, which is sometimes called, Benedict's solution changes colors in the presence of glucose. However, Benedict's solution only works when you heat it up to a certain temperature. Now, in this example, you see them using a flame, but what I probably did was for you was put it into a microwave. And after a couple seconds, the Benedict's solution, if glucose was present in the surrounding environment, should have changed colors. And lo and behold, I believe that's probably what you saw, or what you should have saw, was that scene, I should say, is that what was initially a nice blue color actually turned to this kind of copper or tan or brown color after it was heated. And because that color change occurred, that was indicative that glucose was present in this water surrounding our cell, and as a result we can conclude that glucose was a small enough molecule to diffuse outside of this selectively permeable membrane. Okay, that's part one. So here we are once again. This is a summary. Iodine, which way to diffuse into the cell because the inside of the cell turned blue black. Starch did not diffuse because the water outside of the cell did not turn blue black. Water got into the cell by diffusion because the cell got bigger, and glucose was able to diffuse out of the cell because when we took that glucose indicator solution and we heated it up inside the microwave using the water that we took out of the cell's environment on the outside, it changed colors. So that's part one. Moving on to part two. In part two, maybe you cried a little bit because of the onions, right? We prepared a wet mount, which was all learning target 3.2 was getting you ready for. See here, there's a purpose for these learning targets that we're doing. You made that onion cell. You initially started with tap water, then you added salt water using the paper towel on one side, and then you added distilled water, right? And you should have observed some changes in the cells. Basically, when you're looking at tap and distilled water, the cells looked more or less normal. However, it was a little harder to see, but tap water cells are not as plump or as full as they are when you use distilled water. But salt water is very, very clear here. And when we added salt water, at least what you should have seen to your onion cells, is that the salt water shriveled up. And you can actually see here, these is the cell wall, and inside here is the cell membrane, and this is the cytoplasm. You may even be able to see a nucleus here, right? Some other organelles. But it's evident here that from this picture where there's tap and distilled water, or we should just probably say we started with tap water, right? When we added the salt water solution to the onion cells, they lost water, which means that osmosis occurred. And osmosis is just a special word for what we say for the diffusion of water. And which direction did the water go? Well, the water left the cell. How do we know that? Because it shriveled up. The cytoplasm is made primarily of water, and that must have meant that there was a higher concentration of water inside the cell than there was outside of the cell. And we know that nature likes to be in equilibrium or balance, so how did the cells cope with this or deal with this? Well, they diffused water by osmosis to the outside of the cell, the environment. Now let's say we wanted to reverse this, and you did reverse this when you added distilled water. When you added distilled water, what happened is the cells had now a higher concentration of water on the outside of the cell than they were on the inside of the cell, so the water diffused into the cells by osmosis, and you should have seen these once kind of shriveled up cells returning to their normal size using distilled water. And that's because distilled water is 100% pure water. The salt solution you were using is 90% water and 10% salt. And the cells on the inside, we could say, well, maybe they were about 95% water. That's just a guess. I'm not totally sure. And as a result, you can see that movement of water across the cell membrane. So just as a review, when we added salt water, there was a concentration of water was higher inside of the cell because the surrounding environment was 90% water. We'll say the inside of the cell was 95% water. So the water diffused out and the cell got smaller. However, when we rinse those cells with distilled water, there is now a higher concentration of water surrounding the cells, the onion cells. So the water moved from a higher concentration to the inside of the cells by a lower concentration. The direction of osmosis was inside of the cells, and the cells got bigger. I know that's probably really complicated. That's a lot of words. So if you need me to go over and sit with you and explain it, I'm more than happy to do that. But this is a place for you to go and practice those terms yourself. Before you take the verbal quiz, you probably want to familiarize yourself with these words. Diffusion, osmosis, cell membrane, selectively permeable. Molecules, you should know what those are and how their different sizes are, like glucose is a small molecule and starch is a large molecule. You should know what a solution is, which is just a combination or a mixture of substances. Typically, like glucose and water would be a solution, or iodine and water should be a solution. And lastly, you should be familiar with the term indicator. And remember, an indicator is a substance that tells you whether or not something is present. So if iodine is an indicator, it tells you whether or not starch is present when a color change occurs. Glucose indicator solution is an indicator that changes colors when glucose is present. Okay, so that is a review of the New York State Diffusion through a membrane lab. Hopefully that is helpful to help you understand what was going on in your lab so you can answer the analysis questions or better prepare for the verbal quiz. I think that's it. Let me check the 0 Yes, that's all. Okay. See you soon. Take it easy.